In this lab experiment, kimchi was made to demonstrate the use of microbe fermentation to produce a food product. The recipe for kimchi called for cabbage, kosher salt, garlic, ginger, sugar, red pepper flakes, radish, green onions, fish sauce, and water. After the ingredients were prepared and combined, they were poured into a mason jar, which was lidded and tightly closed. The recipe I used instructed for the jar to be left at room temperature for 2 to 3 days while fermenting occurred. Further instructions suggested that the jar be briefly opened each day to release buildup of gases. According to the Review of Diversity of Microorganisms in Global Fermented Food and Beverages, fermenting species responsible for kimchi are Lactobacillus and Pediococcus initially, followed by active fermentation of Leuconostoc, Weissella, and Tetragenococcus. Taking a closer look at our bacteria, Lactobacillus and Leuconostoc families are considered lactic acid bacteria. These are obligate fermenters that produce lactic acid as an end product. Lactic acid from these bacterium's metabolism contributes to the rich flavor of foods. They are also responsible for fermenting, as well as serving as a preservative purpose. In kimchi, these bacteria are mainly fermenting the cabbage and various vegetable components of the mixture. These bacteria are naturally found in the ingredients, and no live culture is needed to induce fermentation, like in yogurt. High salt content from the kosher salt makes an unfriendly environment for most other bacteria. This gives the lactic acid a chance to thrive. Acidifying the environment also prohibits competitive bacterial proliferation. Leaving the mixture at room temperature encourages desired bacteria to proliferate and perform their job. Fermentation is a process used by these bacteria because they are not capable of respiration, at least not how we perform it. Generally, fermenters do not possess an electron transport chain, so they must process nutrients by an alternate means. In order to generate ATP, the bacteria must perform glycolysis phosphorylation at substrate level. NADH is oxidized to regenerate NAD, with use of an organic compound, pyruvate, to act as a terminal electron acceptor. This perpetuates the cycle of glycolysis. Because pyruvate is used as the electron acceptor, lactic acid is the end product. Due to the breakdown process of glucose, carbon dioxide gas is also released by the bacteria. Going back to the kimchi recipe, cabbage and assorted vegetables are being fermented by naturally occurring lactic acid bacteria. I didn't find anything to confirm it, but I think that sugar was also added to provide a glucose substrate for the desired bacteria. Salt was used to make the food mixture inhospitable to competitive or potentially harmful bacteria species. Fish sauce is also a fermented food. Though no live culture addition is necessary, this ingredient may assist in desired habitat and food product results, and additionally possibly for taste. Pepper flakes are used to introduce flavor, as well as having antimicrobial properties towards undesired bacteria. Water assists with the proliferation and promoting an anaerobic environment. A tightly closed jar also promotes an anaerobic environment. The types of vegetables used are probably not only selected for microbe fermenting properties, but also for desired taste. And actually, now that I have learned more about the process in detail, I realize that rinsing the vegetables thoroughly before mixing may have inhibited fermentation process by removing the desired microbes. Due to the behavior of the fermenters, it is likely that the mixture became increasingly acidic as days passed. Small bubbles could also be observed traveling up the mixture. More bubbles arose as the days went by, which indicates carbon dioxide release, which would also assist in lowering overall pH. Oxygen is not consumed by the bacteria, so O2 shouldn't have decreased. As far as success in making the kimchi goes, that is going to remain undetermined. I have never made kimchi before, and the smell from my batch is quite overwhelming. As far as changes made, it is not likely I will make this food again, but I wouldn't rinse them as thoroughly. I don't want to remove the bacteria responsible for the fermentation of this product. Additionally, I don't trust my fermented food making abilities. I'm not risking getting sick from this stuff, not even in the name of science.